Hi guys, it's Mr. Y. Today we're going to continue with our genetic unit talking primarily about Punnett squares. So the learning objectives for today, learning how chromosomes are related to genes, how the words genes and alleles are connected, and how dominant and recessive alleles interact, learning how to use Punnett squares and how probability relates to Punnett squares, and learning about the terms genotype and phenotype. So let's get started. So all species, whether you're a human, whether you're a dog or a cat or a cow or a tree, all species have a set number of chromosomes. That is to say, within the cells of the body of those species, they have the same number of chromosomes. The only spot this wouldn't hold true would be in like the sex cells, the sperm and the egg. Or seeds in certain cases. Now, for things like dogs, for instance, wild dogs, wolves, they have somewhere around 70 to 80 chromosomes. Cats, in their regular cells, they have 30 to 40 chromosomes per cell. Yeast cells, even simple yeast cells, have 32 chromosomes. Carp, which is a type of fish, has 104 chromosomes in its body cells. Algae, like pond scum, has 148 chromosomes. So what you have to remember is that the number of chromosomes does not connect necessarily to the complexity of the life form we're talking about. Humans, we have 46, which you should have learned about in life science in 7th grade. But even something as simple as a pond protist, which is a very simple one-celled organism, can have as many as 15,000 chromosomes in its one cellular body, and that's it. So the number of chromosomes, or the amount of DNA, does not necessarily connect to how complex the living thing appears to us from the outside. Now, most of these chromosomes will form into pairs. So we say humans, while they have 46, will often have 23 pairs. It's the same thing. And the overall way you get the 46 chromosomes is because you inherited 23 of these chromosomes from your mother and 23 from your father. 23 from the sperm cell, 23 from the egg cell, thus giving you 46 overall. And of these 46 chromosomes, 44 of them, that is to say 22 pairs, are what we call autosomal, meaning they have no influence in any way upon the gender of the organism, in this case humans. So 2 of these chromosomes, however, are what we call the sex chromosomes. These determine the offspring's gender, whether it's going to be a male or a female. So the way we recognize this is that in humans, a female has 2 what we call X chromosomes, whereas a male has an X and a Y chromosome. And again, you'll note that in these pairs, one side had to come from the mother, one side had to come from the father. So in this case, I've labeled the bluish colored chromosomes, the X ones, come from the mother because the mother can only give an X chromosome, whereas the X or the Y came from the father. And you'll note again that the father is actually the one who determines the gender of the child. So here is the karyotype, that is to say all the chromosomes for a human being laid out on the right here in nice pairs. They have them numbered pairs 1, 2, 3, 4, 5. And they go all the way up to pair 22, and then here's the sex chromosomes, and then I've magnified them here as well. So these are the sex chromosomes. The others are all called the autosomes. Right there. Now, you'll note from this karyotype in the top right, what's the gender of this person? Well, this would be a male. The way we know that is because they have an X and they have a Y. Notice the Y is not really Y-shaped. It's really just a very, very, very small X. It looks like parts of the DNA have been lost. So you can see like there'd be usually more parts of the DNA that's somehow not there. That does mean that in fact females tend to have more DNA than males because they have 2 X chromosomes. Now, onto a gene. Now, we covered this in the last lecture, but I want to make sure we go over it again. A gene is a specific segment of DNA that codes for instructions for specific proteins, and that these proteins can gradually influence an organism's trait if there's a lot of them. So if it builds a certain type of cell, like a skin cell, it can gradually build enough skin cells using that same protein again and again and again to get a specific trait concerning that skin. So a trait is a characteristic, like say red hair or blue eyes, that an organism can pass on through its genes. Keep in mind, traits are in our version of this are always genetically based. That is to say, they are always passed on. Some people will refer to traits as things that are inherited throughout their lifetime. Those are not passed on. But for us, when we say traits, we mean things that can be passed on through DNA, through an organism's genes. And let's clarify the term alleles. Alleles are different forms of the same genes. So for instance, freckles. Now, this is a very simple one, because you either have freckles or you don't have freckles. So there's two different alleles for the same gene that produces freckles on the skin. There's the dominant allele, which is, I'm just going to use the letter F, for freckles, and the recessive allele, not for freckles, or for no freckles. So you either have it or you don't in some cases, and with other cases, like with hair color, there's just different degrees of hair color. Another good one, that's a very simple one, widow's peak. The widow's peak is the point of the hairline. You can see this here in this picture here. It has very clear, distinct widow's peak. The gene for widow's peak is dominant. We're going to use just capital W. The gene for no widow's peak is recessive, lowercase w. Now, we'll talk about dominant and recessive more in a minute, but keep in mind, if it's dominant, it's always a capital letter. If it's recessive, it's always a lowercase letter. So the dominant allele always appears in the organism when it's present. It's a trait that will always show up. All it needs is for one gene to have the dominant. Like if you have one gene for freckles, you're going to show freckles. If you have one gene for widow's peak, you're going to have a bit of a widow's peak. It's dominant, meaning it always takes over for the expression of that trait. The recessive allele, on the other hand, is usually hidden, and it's shown in lowercase letters, like I said. And so when the dominant is present, the recessive allele is always hidden. The only way a recessive allele appears in terms of traits is when it's by itself, when there is no dominant allele. So remember, every organism has at least two genes coding for the same basic proteins, but they can be different alleles from each other. So for instance, this individual has a dominant allele for freckles and a recessive allele for no freckles from the same gene. Now, what would this person actually show? What would their physical expression show? Well, because it has both of them, the dominant one takes over. And remember that in this case, dominant is for freckles. So their phenotype, the physical expression of this, would be them showing freckles, because that's the dominant allele. The recessive allele, again, would only show if it was by itself, if you had two lowercase f's in this case. So this person with capital F, lowercase f, they would show freckles. That would be what we call their phenotype, their physical manifestation of these characteristics. Two capital F's, what would they show? They would also show freckles, because again, you have both dominant traits for freckles, both dominant alleles, for freckles. And what if we had an individual who had lowercase f for their alleles? What would they show in their phenotype? They would show no freckles. This is the only time the recessive allele is expressed, when again, it's by itself. You have a lowercase f and a lowercase f. There's no allele for freckles to be dominant. So with this type of situation, you can see it breaks down to a pretty nice, neat, straightforward system, to where the genotype, that's the genetic makeup, so you can put down genetic makeup, has three possibilities. You either have both dominants, that's this one, both recessive, that's this one, or a mix of the two, that's this one in the middle. The phenotypes, again, also have three possibilities. You either show freckles because you have both dominants, you either show freckles because you have one dominant, one recessive, or you show no freckles because you have both recessive alleles in this case. So there's really only three possible outcomes with situations like this, and this is not always the case. These are simplified examples, but they do work for the basics. Now, terms you're going to have to learn. These. When it's two dominant alleles, we say it's homozygous, meaning the same. Dominant. Homozygous tells you they're the same. Dominant tells you they're both capitalized letters. When they're both lowercase letters, they're still homozygous because they're both, again, the same. But now they're homozygous recessive. Recessive meaning both lowercase letters. When they are a mix, one dominant, one recessive, it is called heterozygous. And for the record, heterozygous always puts the capital letter, the dominant allele, first. You would never see the lowercase come before the capital. That's not standard notation for this. Heterozygous is always dominant because the dominant has to be there because it's heterozygous. So if it's heterozygous, the dominant letter goes first, the lowercase letter comes afterwards. So this is what we call the genotype, the genetic makeup of the individual, based upon the alleles that they have and how they pair up. Again, one for the mother, one from the father. The phenotype is the physical expression of these traits that are inherited genetically. Again, we're not talking about things like scars or language abilities. These tend to be inherited from the environment. We're talking strictly about traits that are genetically based, things that are inherited through the DNA. So, for instance, we can extend this idea to predict what will happen in certain cases. So, for instance, if we had a parent who was homozygous dominant, that is to say, two capital Fs, 4 freckles, and a second parent who is homozygous recessive for no freckles, what are the odds of their kids having freckles? And we can mathematically figure that out using a thing called Punnett Squares. This is what a Punnett Square looks like. It's named after a mathematician who originally designed this. So you have one parent here on the top. The other parent gets listed here at the side. And notice what they've done. They said, well, the first parent can either give a capital F or they can give the other capital F. The second parent can either give the recessive lowercase f or the other recessive lowercase f. Now it's just a matter of filling in the boxes. And the way you do that is you bring one down and you bring one over. So for instance, we can use this to figure out the odds of any of their kids having freckles. And notice what happens. In every case, you end up with the same results. A capital F from one parent and a lowercase f from the other parent. Meaning that in 4 out of 4 situations, the offspring is going to have freckles. Meaning the chances are, 100% of their time, any kids they produce, parent 1 and parent 2, they're going to have freckles regardless, no matter what. This is the power of Punnett Squares. It allows you to make predictions of the likelihood of something happening between 2 parents, if you know the genetic makeup of those parents, the genotype of parents. Now, let's take this through another practice problem. What would the cross of 2 heterozygote parents look like? Again, heterozygotes means one capital, one lowercase. So, you draw the Punnett square, like so. Again, one parent goes at the top, and one parent goes at the side. I'm going to put this one all the way down here. And you fill in for all the possibilities. So I'd like you to pause the video and see if you can fill this one in on your own. Again, all it is you bring it down and you bring it over and you do the same for the others. You bring it down, bring it over, and fill in. Tell me, what are the odds of either producing a kid with freckles or a kid without freckles? So you'll note in this time we got a different result. How many of the kids here, in this case, what's the probability of the kids having freckles? Well, in this case, here, 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 freckles. But right here, no freckles. So this is what you always see with the heterozygous cross. 1/4 of them are going to be homozygous dominant. That is to say, they have both dominant genes for freckles. 2 out of 4, or 1/2, are heterozygous. Remember, heterozygous is also dominant. Those are going to produce kids with freckles as well, but they're only heterozygous dominant. The recessive allele is still right here. And then 25% of the time, 1 out of 4, you have a kid with no freckles. So you get the same ratio of 1 to 2 to 1 every time you do a heterozygous cross. Doesn't matter what the gene is, it always comes up with this 1 to 2 to 1 ratio. 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive offspring. Always the same ratio for any hybrid cross you do, and hybrid is a loose form for heterozygous individuals. And the reason we discovered this was through a monk named Mendel, who was working in gardening, actually. And he was studying specifically pea plants, and he was always fascinated by why certain pea plants exhibited one trait, like, say, a purple flower, while others exhibited another trait, a white flower, and they were not equal in terms of how often they came about. So after studying this for many, many years, he was actually able to predict the likelihood of getting, say, a purple flower versus a white flower, or a yellow seed versus a green seed. And he was able to mathematically figure this out long before Punnett squares were ever invented. So let's look at a few of the traits he looked at, specifically the seed shape. Round, which is dominant. Everything on the left side here, these are all the dominant guys. Versus, so we have round versus wrinkled. These are the recessive. So round is dominant, wrinkled is recessive. And let's look at the flower color. In this case, purple is going to be dominant to white. So for the letters, we always usually go with the dominant trait as an idea for how to make the letters. But in the case of seed shapes, we're going to go with R for round seed, lowercase r for wrinkled seed. Capital R is dominant, lowercase wrinkled is recessive. So if I have two pea plants, one that's heterozygous, one that's homozygous recessive, what is the likelihood of having round seeds versus wrinkled seeds? Again, you could use a Punnett square to figure this out. You take one parent, you put them across the top. Capital R, lowercase r. You take the other parent, the homozygous recessive parent, you put them on the side. It doesn't matter which parent goes on the top and which one goes on the side, as long as you're not crossing the genes between the top and the side. One parent on the top only, one parent on the side only, and it works out fine either way. So you then fill in the Punnett square, and you get the following results. Capital R, lowercase r, and then lowercase r, lowercase r. And then you continue. And you notice, well, what's my ratio here? What's my ratio of round to wrinkled? I would have 50% rounds, 2 out of 4, here and here would be round, and the other 2 out of 4 would be wrinkled. So 50% of the time, you would produce a plant that would produce wrinkled seeds. The other 50% of the time, you would produce a plant that produces round seeds. So it's a 50-50 chance in this crossing. Let's try a slightly different one. Here's homozygous dominant and heterozygous. What's going to happen now? Well, you make your Punnett square. Again, one parent goes on top, one parent goes on the side, and you fill in. Notice this way, we get our 1 to 2 to 1 ratio. We have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. But in terms of round versus wrinkled, well, here's a round, here's round, here's round, and here is also round. So there is no chance for producing any type of wrinkled seeds in this case. 100% of the time, this is going to come out and produce a plant that produces round seeds. Now let's switch up the traits. Let's go on to flower color. Again, Mendel noticed that flower color could either be purple or it could be white. There was no mixing of the two. So purple, he figured out mathematically, was the dominant one because it showed up the most often, whereas white, we're going to use lowercase p, was the recessive. So capital P for purple, lowercase p for white. So what's the likelihood of having a purple to white flower if you have a homozygous recessive parent and a heterozygous parent? Again, you got to remember what these terms mean. Homozygous means the same. Same in terms of being recessive means both of these are going to be lowercase p's. Heterozygous means you have one of each, one capital, one lowercase. Now you can proceed. You take the homozygous recessive here on the top, the heterozygous here on the side, and you do the Punnett square. The results? Well, how many are going to be purple? 50%. These top two. How many are going to be white? 50%. The bottom two. So 50% purple flowers and 50% white flowers in this situation. Again, the Punnett square is very, very useful for making predictions about these traits where you have either one or the other. They don't always work this nice and simply, though. Like I said, with things like hair color, you can have a lot more than just two options. It's not just blonde or brown. There's more options than that. So this is a nice example of how basic genetics works, but it's not 100% accurate for everything. But keep in mind, there's more than one trait that makes up an organism. So this is what we really have to compare with the realities of life. Here I have a pea plant that has round seeds and purple flowers. It's homozygous dominant for both. Here I have a pea plant that is wrinkled in seeds and white in flowers. And I want to know, okay, if I cross these two, what's the possibility of getting, say, for instance, a plant that produces wrinkled seeds with purple flowers? Now, to do this Punnett square requires a bit of thinking, because you can't just do it as a 2x2 box anymore. Because you're dealing with two separate traits, the box is going to increase, and it increases a lot. But if you're smart, you'll notice that this guy can only give you a capital R and a capital P. This guy can only give you a lowercase r and a lowercase p in any situation. Meaning, no matter how many times you cross this, 100% of the time you're going to have nothing but round seeds with purple flowers. You're only going to produce heterozygotes. However, this situation you have to do the box for, because you can get lots of different combinations. And the way this would look, well, when you do more than one allele, the box grows by a factor of 4, because this is what you would have to do. You would have to play out every situation, because you could get a capital R, capital P, capital R, lowercase p, and then, lowercase p, capital P, and then etc., etc. And then you'd have to fill in the box. So notice, I add in one allele, and the whole thing goes up by a factor of 4. And you get lots of different outcomes. You have round and purple flowers. Down here you have wrinkled and white flowers. And then you have lots of different scenarios going in between. But there is a pattern here. But this is why we typically don't do this for more than one trait, because if I was to do this for 3 traits, it again would go up by a factor of 4 easily. So we usually only use Punnett squares for 1 trait, sometimes 2. That's usually where it stops at.